Okay, part three. We'll get going again with the second part of this morning lectures. So, for the first 55 minutes or so, I gave you an introduction and covered really our understanding of the clinical presentation of Alzheimer's disease and our current understanding of what drives that, the causes of Alzheimer's disease. What I'll move on to for the second part of this morning's lectures are how we are, as a research community, trying to develop novel therapies for Alzheimer's disease. Treatments that can actually modify the disease, slow it down, stop it, maybe even stop it ever occurring. And this has been a huge area of activity for decades now. There's a clear understanding that Alzheimer's disease and related dementias are a major challenge to healthcare systems across the world. Actually, there's been talk of dementia in particular as being an existential threat to healthcare systems like the NHS. If you're thinking of an increasing burden of individuals with dementia, treating them over the course of many years, over the development of the disease, this has the potential to bankrupt systems like the NHS and have a severe impact on other healthcare systems, insurance-based systems, and so on and so forth. So what I'll talk about are some of the ways in which this is being approached. And actually, the majority of drug trials really have used the amyloid cascade hypothesis as a starting point for trying to develop new therapies. So I'll talk about some of the preclinical development and clinical trials that have occurred in drug discovery in Alzheimer's, and cover some of the pathways being targeted, basically building on what I discussed in the first part of the talk. First part of this morning's lectures, and I'll discuss some of the challenges that are being faced in drug discovery in Alzheimer's disease. In a way, I can summarize the Alzheimer's drug discovery field by saying that we've had dozens of clinical trials, and not a single one of them has worked in terms of a disease-modifying therapy. You're looking at hundreds of millions, if not billions, of dollars, pounds, euros, and yens being spent in drug development over the past 30 years, and there is not a single licensed drug to improve the lives of people with Alzheimer's disease. And that gives you an idea as to exactly how challenging this is. If you want another way of talking about this, if you look across the pharmaceutical sector, headlines come on a fairly and somewhat depressing basis of drug companies pulling out of Alzheimer's research, basically because they're scared by it. They find it very challenging. There are a number of reasons for that. I would view it as short-sighted. I would say that, but pharmaceutical companies are really struggling to develop compounds that target Alzheimer's disease. I will actually end on a positive note, and I'll use the examples of multiple sclerosis and of a very rare form of motor neuron disease called spinal muscular atrophy as examples of central nervous system disorders where actually progress has been made. Clinical trials have been carried out and disease-modifying compounds have been developed. These actually bring their own problems, and I'll mention that very briefly at the end. So, in terms of what I'll cover, what is the challenge? What are the drug targets in Alzheimer's? The two key areas that I'll talk about, and these are the areas where most activity has been over the past several decades, is targeting amyloid beta and targeting tau. And as I said, I'll end on a bit of hope for the future. So the underlying problem in Alzheimer's disease is neurodegeneration. It is catastrophic and widespread neuronal loss. Efforts by the Pharmaceutical Centre and by academic laboratories across the world is basically focused on trying to understand what causes this and then finding ways to intervene in that, to try and block the events that lead to neurodegeneration. And to really understand the way in which this has been approached and to understand some of the pitfalls that have occurred because of that, you need to have at least an overview of the drug discovery process. And so what you have, and there's a number of very good reviews on this. This is taken from a Nature Reviews in Drug Discovery paper. I'll post this on Blackboard this evening, so if you're interested in learning more about this, you can read a bit more about it. And basically you go from initial target validation and drug candidate discovery, identifying where you're going to intervene. What is the molecular target that you're going to hit, that you're going to try and modulate? And this involves actually a huge amount of very early stage research involving the characterization of particular proteins, enzymatic processes, physiological processes within cells and within organisms. This is predominantly carried out now by academic laboratories and has been for a number of years now. This then leads on to preclinical development. When you think you have a molecular target that you can modulate, you then start thinking about, okay, we think we can change this, what's happening here, try and change the way A-beta is being produced, for example. We'll then develop candidate drugs which target that process and scale them up in preclinical development. A lot of this involves cellular modeling, modulating a process in cell culture and cell culture models for disease, and moving into animal models. These involve in vivo toxicity, safety, and efficacy testing, making sure that the compound that you've developed or the biological approach that you're taking targets what you think it is target, proving that you can engage the target and that it has the consequence that you think it is going to have, perhaps reducing the production of A-beta. As we know that overproduction of A-beta, you have an extra copy of APP, you get Alzheimer's disease. The idea is if you reduce the production of APP, then of APP and of A-beta, then you can reduce the risk of developing Alzheimer's. So that's an example of how this process works in terms of the sort of logical steps. Finally, once you've done that preclinical development and you have a compound that you think is safe in animal models, does what you think it should do, and seems to have a benefit in disease models, you then move on to clinical studies. These are divided up into phase 1 clinical studies, with the early stage where you're checking safety, checking that people don't become worse when you have the drug, and then moving on to testing whether or not it actually has a clinical benefit. This is summarized in this slide here. So phase 1, you're assessing safety, testing the doses, making sure that you're not causing harm with your compound. If you can get past this, these tend to be relatively small in scale and relatively small in cost, somewhere around $15 million, although those costs are increasing. A drug which I'm very familiar with from my work in Parkinson's disease, the phase 1 trials for that, which are ongoing at the moment, are costing about $95 million. This gives you an idea as to the scale of cost for candidate drugs coming through. Phase 2, where you're testing whether or not actually the drug does what you hope it will do. Does it have clinical efficacy? Does it alter the clinical outcomes, the markers that you have for that disorder? So, for example, in Alzheimer's disease, you would want to see a reduction in the rate of progression, the rate of cognitive decline being reduced. This also involves readouts of target engagement, proving that the drug gets to where you hope it is going to get to and actually alters the process that you're looking at. For example, in Alzheimer's disease, you may take cerebral spinal fluid, take a spinal tap, measure CSF A-beta, and show that you're reducing the production of A-beta. This typically includes somewhere between 100 and 300 patients, so individuals who have the disorder. The phase 1 trials are focused on healthy controls, not on diseased individuals. And the cost is somewhere around 23 million. Actually, for Alzheimer's disease and other neurodegenerative disorders, it's considerably more. Part of the reason being that many of the readouts take a long time to measure. So you have to look at cognitive decline over the course of maybe a year, maybe 2 years. That significantly increases the cost. Finally, you have phase 3 trials, and these are the big ticket events. They are very costly and much larger scale. They're usually placebo-controlled and double-blinded, so the individuals taking part in the trial have no idea who's getting what, a placebo or the real drug. And actually the people giving the drugs, the consultants and the medics involved in the study, don't know whether they're giving a placebo or not. And so their evaluation of the clinical readouts is unbiased. And this is really the gold standard for clinical testing. This also allows you to confirm safety and efficacy, so it sort of builds on the previous 2 phases. These are very expensive and they're very large scale, often involving over 1,000 people and costing upwards of $86 million. That's an average across all types of medicine. For Alzheimer's and other neurodegenerative diseases, the cost is significantly more. What I'd focus on, what I'd like to draw your attention to rather here, is the failure rate. So about 40% of drugs fail at this initial phase 1 stage. When they're given to patients, actually the response of those patients is deleterious. So it's causing harm, and the trials are either immediately stopped or are halted at their conclusion. It's realized that the drug is potentially damaging, and so it won't progress. Phase 2, failure rate is over 50%. 64% of drugs fail at phase 2. Phase 3, these major large clinical studies, 38% failure rate. This is taking a conglomerate of I think it's about 10 years worth of clinical trial data across all forms of disease. Now, if you sum all of this up, you can see that the chances of taking a drug from phase 1 to a successful conclusion of a phase 3 trial is very, very small. Most drugs that enter clinical testing do not work. And if you also tot up the amount of costs, as I say, this is an average of, across a lot of different disorders, the costs are astronomical. You're looking at the very least, hundreds of millions of dollars. In the case of a lot of drugs that are going into neurological disease testing, you're talking about billions of dollars, and eventually failing. And this actually is the story of Alzheimer's disease drug discovery. The drugs that have been developed at huge cost have all failed. There are a lot of ways in which this has been approached in Alzheimer's disease. I'm going to focus on the disease-modifying therapies targeting amyloid and tau, but actually there are a wide range of approaches that have been taken. As I mentioned in the lecture earlier on this morning, modulating neurotransmission as a symptomatic therapy actually is something which appears to have a small but significant clinical benefit in a minority of patients, normally under 50%. This is not disease-modifying. This is a symptomatic treatment. In terms of disease-modifying strategies, the majority of these are amyloid and tau-based. They're targeting the underlying pathology, targeting the proteins which aggregate and cause, we think, that damage in Alzheimer's disease. Coupled to that, there's a lot of research that's gone on looking at the modulation of intracellular signaling cascades, basically the way in which cells are responding to this damage. Oxidative stress reduction, so it's thought that accumulation of amyloid and tau causes an increase in oxidative stress, and this contributes to cell death in Alzheimer's disease. Mitochondrial targeted therapy, trying to boost mitochondrial biology. Again, this in particular in Parkinson's, actually, there's been a lot of evidence suggesting mitochondrial dysfunction is linked to neurodegeneration. Modulation of cellular calcium homeostasis, the control of calcium levels within the cell is a key component of pathways that can lead to cell death. And so there are a number of therapies which have been developed to try and modulate this in the context of Alzheimer's disease. Increasingly, we're appreciating that anti-inflammatory approaches may have clinical applicability. And actually, a lot of this comes out of the realization of the other part of the pathology of Alzheimer's disease. I talked about tau and amyloid accumulating, but it's important to remember that there's a cellular component here as well. The astrocytes, activated microglia, part of that response is an inflammatory response to disease. And we think that some of that inflammatory response is actually harmful, is actually exacerbating the underlying disorder. And so increasingly, pharmaceutical companies and academic labs are turning to anti-inflammatory strategies. And the potential for disease-modifying symptomatic treatments as well. So trying to improve ways of boosting cellular function to modify disease course. This is a snapshot from last year, I think, of the drugs which are in development for Alzheimer's disease. And if you think back to the slide where I showed you all the costs of all of those therapies, this gives you an idea as to how much is being invested in drug development in Alzheimer's disease. On the outside here, you've got phase 1 trials, phase 2 trials sitting here, and then phase 3 trials sitting in the center here. And you can see that there's a decreasing number as you get closer into the center. The number of phase 1 trials, actually, unfortunately, the pipeline of new drugs coming through is beginning to decrease. What we also see is that this is color-coded, so the filled-in symbols filled in red are drugs which are targeting amyloid beta. The ones which are colored blue are targeting tau. And so you can see quite clearly that the majority of therapies in development are targeting either amyloid or tau. However, there are increasing numbers of drugs which are targeting other forms of issues underlying Alzheimer's. You'll also notice that a significant chunk of these are immunotherapies, and I'll talk a bit more about this over the coming slides. So, as I said, the key pathology underlying Alzheimer's disease in terms of protein aggregation is the accumulation of amyloid plaques, and this is an immunohistochemistry image of the brain of an individual with Alzheimer's disease, showing an amyloid plaque over here on the left, and neurofibrillary tangles, basically filling up cells within the brains of an individual with Alzheimer's disease. And for a number of years, there was actually a very heated discussion within the Alzheimer's community as to which of these was important. Is it the amyloid beta causing the degeneration, or is it the accumulation of tau? And this sort of divided the community up into Baptists, or beta-APPists, and Taoists. The idea that either tau was important or amyloid beta accumulation was important. In reality, the likelihood is that both of them are important. And increasingly, there is an appreciation that amyloid beta accumulation is a key initiating factor in disease. So the initial insult, if you like, is the accumulation of A beta. But this then results in the accumulation of tau. And tau is much, much more tightly linked to neurodegeneration. If you correlate the pathology in the brain in terms of the amyloid plaque burden and the tau burden actually correlates much, much more closely with neurodegeneration. The likelihood is that both of these are important. And I'll go back very briefly to the amyloid cascade hypothesis, which John proposed back in 1992. And the idea that what's happening is that you have A beta accumulating in the brain, causing eventually through a cascade, possibly linking into an inflammatory response partway down, resulting in tau accumulation and then neurodegeneration and disease. So if this is our underlying framework for what causes Alzheimer's disease, we can then pick out individual steps in this process and try and modulate it, try and change the underlying biology. We can try and reduce the production of A beta. We can try and increase the clearance of A beta, reducing the amyloid burden within the brain. We can try and reduce inflammatory responses using a variety of approaches. And finally, we can try and target tau itself. Down here at the bottom. So try and intervene, prevent the accumulation of tau, perhaps try and increase the rate at which it's cleared, or try and modulate the way in which it's causing dysfunction within the brain. And basically, this is where the majority of clinical trials have come from over the past few years. These vary in terms of the precise way in which they've tried to target it. A lot of approaches have been driven by an immunotherapy approach, using antibodies, artificially produced antibodies that target the proteins involved in neurodegeneration. So, for example, solanezumab, produced by Eli Lilly, bapineuzumab, produced by Pfizer, are anti-amyloid antibodies. They're artificially produced recombinant antibodies, which are administered as a passive immunotherapy, which binds to and removes A beta from the body. There are also a number of compounds which are directly targeting the enzymes that produce amyloid beta. So compounds that directly inhibit some of the enzymes, the beta secretase or BACE, or the gamma secretase, remembering that presenilin 1 and presenilin 2 are key components of gamma secretase, targeting these activities and reducing the production of amyloid beta from APP. And so basically what these are trying to do in some way, shape or form is shift the production of A beta. They're either looking to sequester and remove A beta once it's produced in terms of immunotherapy, developing antibodies that bind to and remove A beta and get it cleared out of the brain, or they directly target the enzymatic activities, the beta secretase activity that cleaves APP and the gamma secretase activity shown here that cleave APP, producing the A beta peptide. It's important to note that there are two pathways for APP processing. The other pathway involves processing by an enzyme called alpha secretase, another protease. And so the idea is that if you inhibit beta secretase or gamma secretase, this reduces the processing of APP to produce the A beta peptide and increases the production of the non-amyloidogenic fragments of APP. So put very, very simply and zoomed in on this, basically you're taking APP and you're trying to reduce the amount of A beta that is produced by targeting the proteases that directly cleave APP. And there's actually a lot of preclinical evidence that this can be beneficial, particularly focused on mouse models for Alzheimer's disease, and mouse models which develop quite significant amyloid plaque burdens. This is a paper actually from a few years ago now, back in 2010, showing that modulation of gamma secretase using a small molecule that binds to the active site of gamma secretase and prevents it cleaving APP can actually modulate beta amyloid deposition in a transgenic mouse model of Alzheimer's disease. I won't go through the data in detail here, but basically they used a panel of inhibitors of gamma secretase, and they could prove in vitro in cells that they could reduce A-beta production. This then resulted in a reduction in amyloid plaque pathology in the brains of these mice, which are genetically engineered to overproduce amyloid beta, genetically engineered to overproduce APP, and actually resulted in some clinical benefits. And I say clinical benefits in inverted commas, the reason being that actually the impact on cognitive function in mice when you have overexpression of APP is relatively limited. So these mice develop a huge amount of A-beta pathology in the brain, but what they don't develop is significant cognitive deficits. So this highlights some of the challenges in terms of modeling a complex neurological disease like Alzheimer's in animal models such as mice. There is some truly astounding science that has underpinned a lot of this development. Huge efforts have been made to try and understand the molecular structure of some of these enzymes, some of these proteins involved in Alzheimer's disease. This is a paper from a few years ago, 2015, using cryo-electron microscopy. The Nobel Prize was awarded for cryo-EM a couple of years ago now, to actually get a molecular understanding of human gamma secretase, an atomic structure for this protein. The reason being that if you can get an atomic structure of an enzyme, of a protease, actually you can understand the precise coordinates of the active site, and you can design novel molecules that can get into that active site and modulate its activity. And this is the consequence or the output of studies like this. So this is an atomic resolution structure for the gamma secretase complex. Gamma secretase is an intramembranous proteolytic activity, so it's made up of a number of different proteins. It's actually got 4 component proteins that bind together to form a complex and sits within the membrane. APP basically sits in here, and the proteolytic active site is sitting here in the middle of the membrane. This is a phenomenal feat in terms of structural biology. Purifying a multi-protein complex and getting a structure for it is difficult enough. Purifying a multi-protein complex that sits within the membrane and getting an atomic resolution structure for it is a phenomenal achievement. And actually, this type of advance, knowing exactly what the proteins involved in amyloid beta production look like, is probably where most of the advances are going to come in terms of modulating amyloid production over the coming years. And this is a very active area of research. In terms of the actual compounds that are being tried, this is a compound, a gamma secretase inhibitor that Eli Lilly developed and carried out a phase 3 trial, actually a number of years ago now. This was reported in 2013, a phase 3 trial of small molecule inhibitors for gamma secretase. What this does is it binds to the active site of gamma secretase and reduces the production of amyloid beta from APP. As we'll become depressingly familiar with all of the trials that I'll talk about, this failed. It did not meet its clinical endpoints. There was no cognitive benefit in the individuals enrolled in this trial. Pharmaceutical companies have also been looking at targeting beta secretase. This is a crystal structure of beta secretase, a much simpler enzyme to target. It sits in the extracellular space and is a single globular protein. And so actually we've had a crystal structure for BASE for a number of years now. What that has allowed is the development of inhibitors that directly block the activity of BASE1, beta secretase. Beta secretase is one of those enzymes that cleaves APP to produce A-beta. And again, Eli Lilly, who have been very active in this area of drug development, produce an inhibitor that can reduce the production of A-beta in animal models and actually also in humans. This is basically reporting a phase 1 clinical trial. This is now being moved forward into phase 3 trials. As yet, we don't have a clear outcome for this. This is just to show that actually we can use these crystal structures, structural biology, to help inform the development of compounds that are more able to inhibit beta secretase and to have improved pharmacokinetic and pharmacodynamic properties, i.e. getting into the body, being available, targeting the enzymes at the right point and at the right dosage. Again, this is something that has been looked at in a great deal of detail. This is a trial run between AstraZeneca and Eli Lilly for another compound targeting BACE1. And as I say, as yet, we are not at the point where we have a clear idea as to whether this approach will work. One of the key areas where there has been a huge amount of effort is immunotherapy, as I mentioned a few slides ago. And there are a variety of different ways in which you can use immunotherapy in the context of chronic disorders, in contrast to viral and bacterial immunization strategies, to target disease. And actually, both of these have been tried in Alzheimer's. Firstly, you have active immunization. This will be familiar to you from the approaches which are used in treating viral diseases and bacterial diseases. You take an active or a deactivated virus or components of that virus, you inject that into an individual, their immune system is exposed to it, they produce antibodies against that virus, that then allows the body to mount an immune response to it. In the context of Alzheimer's disease, what has been tried is taking an amyloid beta peptide, injecting it into people. The immune system then recognizes that A beta, generates antibodies against it, and causes the idea is that this causes a sequestration of amyloid beta peptide, removing it and clearing it from the system, reducing the amount of A beta within the body. Actually, the phase 3 trials of this, which I'll go on to talk about a little bit, had quite severe side effects. And so a number, and when I say quite severe, actually a number of people involved in phase 2 trials for active immunization in Alzheimer's using the beta peptide developed essentially an inflammatory response within the brain, an encephalopathy that resulted in death. And immediately the trial had to be halted, and the drug development in that area stopped. And this highlights some of the real costs of carrying out clinical trials like this, that actually sometimes they can result in patients dying. That resulted in a switch to passive immunization, the use of artificially produced antibodies that are themselves a drug. These are biological treatments, biologics, that can be given to people and reduce the possibility that you'll get a sort of auto-inflammatory immune response that causes damage within the brain. And this really is where the community is focused over the past decade or so now. Again, a number of phase 3 trials have been carried out. Solanezumab for mild to moderate Alzheimer's disease, phase 3 trial reported in 2014, and 2 phase 3 trials run by Pfizer and Johnson & Johnson using this approach, taking recombinant antibodies, injecting them into individuals, and looking to see whether that reduced the progression of Alzheimer's disease. Again, the bottom line, no clinical benefit whatsoever. In fact, what is interesting about these studies is that they did engage the target. So there was evidence that they were reducing the amount of amyloid beta in the brains of the individuals who were enrolled in these phase 3 trials. Once everything had been unblinded and the data was being analyzed in terms of cerebral spinal fluid, A beta, and imaging readouts. But that did not have any clinical benefit. And I think that is probably enlightening as to the way in which we approach these disorders, which I'll go on to talk about in a minute. One of the other key ways in which Alzheimer's drug discovery is developed is targeting the latter stages of the amyloid cascade. So actually targeting the intracellular aggregation of tau in neurofibrillary tangles. As I said, tau pathology actually correlates much better with neurodegeneration than amyloid beta pathology in the brain. So if you look at the amount of neurofibrillary tangles and try and correlate that with neurodegeneration, tau pathology is much more closely linked to neurodegeneration than amyloid beta pathology. You can have a lot of amyloid beta pathology, but not a huge amount of neurodegeneration, whereas if you have a lot of tau pathology in an area of the brain, actually you do get neurodegeneration. It's also important to note that tau is involved in a number of other disorders, including rarer forms of dementia and movement disorders such as progressive supranuclear palsy, frontal temporal dementia, and chronic traumatic encephalopathy, which I mentioned. Mike Webster and Muhammad Ali developing dementia pugilistica and chronic traumatic encephalopathy. Tau is is the key presenting pathological hallmark there. So it's involved in not just Alzheimer's disease, but other disorders as well. And so the idea is that you can zoom in on this amyloid cascade series of events and focus in on the latter stages, the bits that we think are directly linked to neuronal cell death and to the dysfunction of neural circuits. And again, there have been some astounding advances in our understanding of the molecular underpinning of all of this. This is a paper that was published a couple of years ago now, back in 2017, again using cryo-electron microscopy as a tool to understand the molecular characteristics of those tangles, the protein that sits within cells. And we now have a molecular level understanding of what those fibrils look like. This is an image of those neurofibrillary tangles. They're made up of paired helical filaments, and we can actually look at those now in at atomic resolution. This is a low resolution EM image of these fibrils. And so there's been a huge amount of advance in terms of the underlying biology. This has led to approaches targeting the aggregation process of tau, so designing small molecule inhibitors of the aggregation process. We can model that in vitro, we can model it in cells. You develop small compounds that prevent tau aggregating and forming these big aggregates. And actually this has gone on to phase 3 trials. This was reported back in 2016. It's a biotech company called Tau RX Pharmaceuticals, and they carried out a phase 3 trial looking at tau aggregation inhibitor therapy in Alzheimer's disease using a randomized controlled double-blind phase 3 trial. Again, no clinical benefit. People have also moved towards an immunotherapy approach with tau, so they're generating antibodies, artificial antibodies against tau, trying to basically suck the tau out of the system and reduce the amount of the potential that it has for aggregation. These are less advanced. This is reported a couple of years ago in the Lancet Neurology, 2017, using a tau vaccine. Actually, this is a vaccine using a virus to overproduce a small fragment of tau, which elicits an immune response, demonstrating that this approach was safe in control individuals, i.e. those who don't have any form of a neurological problem. And this has moved through into phase 2 trials, and so is part of that pipeline of therapeutic tests. As I said, there are a number of other approaches which are being used, and neuroprotective agents, anti-inflammatory approaches. I don't know if anybody saw the BBC documentaries over the past couple of weeks on Parkinson's disease, using a compound called glial-derived neurotrophic factor. This is a neuroprotective agent which is being trialled in Parkinson's. There's also been thoughts of using this in Alzheimer's disease. One of the big problems with that is getting it into the brain. So for those of you who didn't see the documentary, basically they developed a pump that injected this compound directly into the brain, and individuals had to have that engineered into their skulls in order for the drug to be delivered. Actually, unfortunately, it did not have any clinical benefit. This was a small-scale phase 2 trial. Anti-inflammatory approaches are going through that pipeline process, going through phase 1, phase 2 trials at the moment, as are examples targeting mitochondrial biology. There are major challenges in order to blocking the development of drugs that treat Alzheimer's disease. First, and I think probably most important in terms of developing a disease-modifying therapy, is that pathology starts long before initial clinical presentation. And so when somebody reports to their GP saying, I couldn't find my keys, I haven't been able to find my way home, I've forgotten how to tie my shoelaces, I'm beginning to have trouble remembering things day to day, actually by that point, the majority of the pathology has already occurred. The majority of the damage has already happened within the brain. And there's I think probably the balance of probability is now that intervening at that point, by the time people develop cognitive impairment, is probably too late. And that actually we need to be intervening 5 to 10, maybe even 15 to 20 years before that. It's also incredibly challenging having to detect potentially quite subtle changes in the trajectory of degeneration. People get worse over time with Alzheimer's disease, and measuring that rate of decline and seeing whether you can make it slightly less worse, if you like, over the course of a couple of years is actually a challenging thing in terms just in the way in which you design a clinical trial. It's also important to note that actually there is quite a heterogeneous population of patients. And so individuals who may have Alzheimer's disease linked to ApoE genotype may have a slightly different disease process to people who don't have an ApoE genotype. The rate of decline may be more in certain individuals than others. So if you have a group of 30 or 40 patients, you may actually have a clinical benefit in a subset of those patients, but you can't pick it out using the statistical approaches that are used in clinical trials because of that heterogeneity. Finally, and again, this is something that the pharma companies will bring up again and again and again, is that targeting something within the central nervous system is intrinsically incredibly challenging. You have the blood-brain barrier protecting the brain. This protects the brain from infections coming in from the outside and chemicals causing damage to the brain. But it also makes it very, very difficult to get drugs into the brain. You can give someone a pill or an injection, it sits in the periphery, but actually crossing the blood-brain barrier in and of itself is a major challenge. I'll talk next week a little bit in the context of Parkinson's as to how this can be overcome. But that in and of itself is a major obstacle to drug development. Just in terms of the underlying pathology, and these are the slides that I showed earlier this morning, it's really just highlighting that the disease process is occurring many, many, many years before the onset of symptoms, or before the onset of measurable symptoms. And so you've got this accumulation of amyloid beta, and actually of tau as well, happening long before any symptoms are being registered by the patient. And the likelihood is that actually in order to impact on this part, the symptomatic phase of the disease, actually you have to intervene before the symptoms occur. Now at the moment we define Alzheimer's disease based upon its clinical presentation. The only reason you get a diagnosis is because you go to your doctor, you undergo cognitive testing, you might undergo some kind of neuroimaging. Now, if we're saying that actually in order to impact on that disease process, you have to identify the patients before they get the disease, how do you do that? And at the moment, we don't have an answer to that. There are aspects of genetics which can help us there. So for example, there are now clinical trials ongoing where you target people before they develop Alzheimer's, but you know they'll develop Alzheimer's. And you can use a combination of imaging and some biomarkers to get an idea as to who is at greatest risk of developing Alzheimer's. But actually pinpointing, saying this person will develop Alzheimer's at this point in their life, we can only do that for a very small subset of patients, almost exclusively those who develop Alzheimer's because they have an inherited mutation. And increasingly, this is where the pharmaceutical companies are moving. There are a number of large phase 3 trials targeting individuals with mutations, either presenilin-1 mutation carriers for a variety of reasons. There's a small region in Colombia which has a very high density of presenilin-1 mutations. These are across the world very, very rare. There's a couple of towns in Colombia where it is much, much more common. And there is a significant $100 million effort to carry out a phase 3 trial targeting these presenilin-1 mutation carriers, trying to stop them getting disease in the first place, or shifting the age at which they develop disease to later on in life. We can do that in this population because we know that they will develop Alzheimer's disease. They have a mutation, we know that that will cause them to develop Alzheimer's within a 5 to 10 year window. But these are very long and very expensive trials. You're looking at a 5-year duration for a clinical trial. Most clinical trials run for 1 to 2 years and are hugely expensive. This is going to be much more expensive than that. Actually, 96 million is probably an underestimate in terms of the cost. There are also ongoing studies of individuals with other mutations across the globe, and really this highlights that there is an ongoing and global approach to trying to target Alzheimer's disease. What you're increasingly finding actually is pharma companies are collaborating with each other in order to try and reduce the costs of these trials to try and drive drug discovery. And there are also increasing efforts to target individuals who have been identified with high A-beta burden, so have not yet developed any kind of cognitive impairment, but are identified as having high levels of A-beta in their CSF or using brain imaging. Whether these will actually result in any kind of clinical benefit is not clear. The reason we have clinical trials is because we don't know whether or not the drugs will work. The story to date of drug discovery in Alzheimer's is a very depressing one. We've had multiple phase 3 trials at a huge expense. Hundreds of millions of dollars have been spent on these trials, and none of them have worked. And actually, this is one of the things that has driven pharmaceutical companies to move out of the neuroscience drug development space. But I said I would end on a note of hope. And I think there are two disorders that I would point to where there has been substantial progress in terms of developing disease-modifying therapies. First of which is multiple sclerosis. This is a neurological disorder. At heart, it is an autoimmune disease. It's caused by a degeneration of synapses and axonal of axons and an impact on axonal function, caused by an immune response within the central nervous system. This is a drawing from a guy called Robert Carswell, who was the first person to draw the pathology of multiple sclerosis. This was back in the early part of the 19th century. It's a progressive neurological disorder, so it sits within the central nervous system. There are key differences between multiple sclerosis and a disorder like Alzheimer's disease. Multiple sclerosis is, for the most part, episodic, so you have periods of remission and periods of relapse. But actually, there are increasingly appreciated overlaps between between MS and Alzheimer's disease. In particular, the end stage of multiple sclerosis involves progressive neurological dysfunction, and so you end up having a neurodegenerative process. And these are just two examples of individuals affected. Jacqueline du Pré, one of the most talented cellists of her generation, or any generation for that matter, who died of complications of multiple sclerosis and was completely disabled by the disease back in the 1960s. And more recently, Selma Blair, the actor, who has been quite vocal about her diagnosis with multiple sclerosis. The reason I highlight these two individuals is that the prognosis has changed hugely from Jacqueline du Pré's era to the modern day with Selma Blair. So Selma Blair will now be being treated with a number of disease-modifying compounds which are impacting on the disease process. This is just a summary of some of these clinical trials that are being carried out. Many of them immunotherapies targeting immune response within the central nervous system in multiple sclerosis. And these make a difference. These are disease-modifying. They reduce the relapse rate in multiple sclerosis and are extending disease-free lifetime. And so here we have an example of a central nervous system disorder which is very complicated. The underlying processes are very complex in the same way that Alzheimer's disease has a very complex underlying cellular etiology. But actually, the challenge we have now in multiple sclerosis is trying to figure out what combination of the drugs which work best. And so here we have a success story. We have disease-modifying drugs which are making a clinical benefit to individuals with disease. The second example is much rarer. Multiple sclerosis is a relatively common disorder. It affects somewhere of the order of, I think, 80 to 100,000 people in the UK. The second example is much, much rarer, and it's a rare form of motor neuron disease. These are degenerative disorders, particularly affecting motor neurons. So essentially the neurons extending out of the central nervous system, connecting up with your muscles and controlling muscular function. Degeneration of these is catastrophic and will result in disability, so you lose the ability to control your muscles, or you lose your ability to walk. And eventually results in failure of lung function. So innervation of the lungs stops and individuals asphyxiate. This is the consequence of motor neuron disease in the majority of cases. There are rare exceptions where it does not progress to the point where lungs are affected. Spinal muscular atrophy is a particularly aggressive and early onset form of this disorder and actually affects, for the most part, infants and small children. Many of whom do not develop beyond the very early stages of life. So individuals with spinal muscular atrophy is caused by inherited mutations in a gene called SNM1, will not progress from beyond the age of about a year because they're unable to develop the movement that allows them to survive. They can't sit up, they can't crawl, they can't coordinate their movement because motor neurons are already degenerating within their bodies. Now, This is very, very rare. So it affects a handful of children, and quite often a de novo mutation. So they're not inherited per se. They occur in de novo for individuals. Now, what is astounding is that we now have therapies that make a massive clinical impact in terms of life expectancy for those very, very rare cases of spinal muscular atrophy. The approach which is used is a gene therapy approach, and in fact what is done is boosting of a parallel gene to SNM1. Essentially what happens in SMA is that you lose function of SNM1, and that then causes motor neuron degeneration. There is now a gene therapy, nusinersen, which boosts production of a silent gene which sits parallel to SNM1, which replaces the function of SNM1. And this has miraculous impact. Children, infants who were unable to sit up or crawl are now able to meet movement milestones. And really, this is an incredible success story. It's a drug which is making a major impact in terms of clinical outcomes. We don't know how long those clinical benefits will last, but this has gone through a number of clinical trials now and is demonstrated to work. Again, this is a proof of concept that you can go into the central nervous system, you can intervene with a genetically defined molecular target, and you can have a clinical benefit. Unfortunately, the costs of this are huge. Each dose of nusinersen is about $80,000, and the likelihood is that you will need to take that for the rest of your life. NICE, the body in the UK which licenses drugs and recommends them, not licenses drugs, recommends them for use within the NHS, has not recommended that nusinersen is funded by the NHS. So if you have a child who has a mutation in SNM1 and develops spinal muscular atrophy, the NHS will not pay for this drug to be used because it is so expensive. The company which developed this drug is now developing similar gene therapy approaches targeting Huntington's disease, targeting amyloid beta, targeting tau, targeting the prion protein, and I'm almost certain targeting alpha-synuclein in Parkinson's disease as well. Certainly the ones that are in the public domain are prion disease and Huntington's disease, and there's an ongoing phase 2 trial in Huntington's disease using almost the same approach. It's not quite the same approach. They actually use a gene therapy approach to knock down the production of the Huntington gene. We will know within a few years whether this approach will work in much more complex adult onset forms of neurodegeneration. The problem is, if they do work, we won't be able to afford them. And at the moment, I don't have an answer as to how we get around that. But the likelihood is that some of these approaches, and they may be gene therapy approaches, they may be earlier use of the compounds that can target APP production or tau accumulation, will have a clinical benefit in the next 10 years. The problem will be that the costs are astronomical. And if you're looking to treat 600,000 people in the UK with these drugs, you need to find that money from somewhere. And at the moment, there is no answer to that. So just to return to this pipeline in terms of hope for the future, the history of drug development in Alzheimer's disease is one of repeated failure. I think looking at the examples of multiple sclerosis and of spinal muscular atrophy, actually I genuinely, and actually for the first time in a long time, I have genuine hope for the future in Alzheimer's disease in terms of developing those therapies. I think the consequences of that are going to be very complicated. If we do develop drug therapies that work, but only the wealthiest 10% of the population can afford them, that has major implications for the way in which our society functions. I do not have an answer for that, unfortunately, but there is a bit of hope there for the future. This really is just a summary of all that I've covered this morning. I'll be posting a number of papers on Blackboard relating to Alzheimer's disease, and I just checked through the library. Actually, you can access my textbook online for free, which is somewhat disappointing for me because I get 10% of all the profits from that book. But you can actually access it for free through the library. You can download a PDF. So if you want to go, you know what, I'll post the link for that on Blackboard as well. I would also highlight, I think I do get some royalties from the library having to pay for the access, by the way, so it's not entirely altruistic. I would really encourage you to look at Alzheimer's and other neurological disorders more broadly. These are some examples of literature and film portrayals of Alzheimer's disease. These are fictional in some cases. It's been highlighted that King Lear is actually a very good representation of dementia in later life. This is the film about Margaret Thatcher and about Iris Murdoch. These are actually quite good representations of the disease process in Alzheimer's disease, but I'd really encourage you to engage with some of the material that the Alzheimer's Society and Alzheimer's Research UK provide. These will give you an insight as to exactly how devastating dementia can be, and will give you an idea as to why it is so important we need to develop new treatments. Thanks very much. Of course, I'm more than happy to answer any questions if you want to come up and ask them, and I'll be posting the YouTube videos later today. And see you next week for Parkinson's disease.